Good evening. Let's try it again. Good evening.
Good evening.
Thank you. I'm Sarah Morris, the John M. Kingsbury Executive Director of the Shoals Marine Lab, and I would like to thank everyone for joining us tonight for our sixth Rock Talk of the 2026 season. For those of you joining us online or who may be new to the lab, we are the largest and oldest marine lab dedicated to undergraduate training in the United States. The lab is operated by the University of New Hampshire and Cornell University. Our seminars continue to be named Rock Talks because back in the day, before we had walls for many of our buildings, many of the talks actually happened outdoors, literally on the rocks. Now we move inside, which makes it a little bit more comfortable on days like today when we've had as much rain as we have. We have migrated indoors, and this provides an opportunity for many of our members to come together to hear about different aspects of marine biology or environmental science as part of our island community. In addition to our guests joining us remotely tonight, we have 3 classes on the island: marine mammal biology, anatomy and function of marine vertebrates, and R by the Sea. For those of you who don't know, R by the Sea is a hybrid course. This is their first week on the island. Nice job. In addition to our students taking our 2-week classes, we're also joined by our 10-week research interns in the Shoals Undergraduate Research Group, affectionately known as SURGES. We're excited to have everyone both in person and online tonight for this seminar. The format will be about a 45-minute talk followed by 15 minutes of Q&A. For anybody who is online, please use the Q&A button at the bottom of your screen. For those of you in the room, we'll be coming around with the microphones so that the people online are able to hear the questions and have them be answered. Tonight's speaker is Dr. Matthew Leslie, better known as Marine Mammal Matt here on Appledore. Dr. Leslie is an assistant professor in the biology department at Ursinus College. He received a BS degree in zoology from Oklahoma State University. Prior to attending graduate school, he managed the Sackler Institute for Comparative Genomics at the American Museum of Natural History in New York and managed the Conservation Genetics program for the Wildlife Conservation Society. He then went on to graduate school and earned his MS and Ph.D. degrees from Scripps Institution of Oceanography in affiliation with NOAA's Southwest Fisheries Science Center. He has served as a Secretary's Distinguished Postdoctoral Research Fellow in the Smithsonian Institution's National Museum of Natural History and National Zoological Park, a Science and Technology Policy Fellow with the National Climate Adaptation Science Center within the USGS, and a visiting professor of conservation biology at Swarthmore College. Matt has also served as an invited participant to the scientific committee of the International Whaling Commission. He's received research funding from the Sussman Foundation, UC-Mexus, the American Museum of Natural History, National Science Foundation, National Museum of Natural History, Smithsonian Institution, NOAA Southwest Fisheries Science Center, and the U.S. Marine Mammal Commission, among others. This coming spring, he will be spending in Panama as a Fulbright Scholar in Science, Technology, and Education. His scientific research combines conservation-driven studies of mammals. Specifically, Matt employs a unique combination of fieldwork, genomics, and natural history specimen-based inquiry to uncover natural patterns and processes of evolution, ecology, and population biology. The goal of his work is to provide information for targeted actions that conserve and recover wildlife. Matt has conducted field expeditions, I'm very jealous, in Madagascar, Cambodia, the Western Hawaiian Islands, Chilean Patagonia, the Bahamas, the Gulf of Mexico, Oman, Australia, Panama, and South Georgia Island. This evening, he will be presenting combining student experiential learning with research and conservation of tropical non-migratory baleen whales. Please join me in welcoming our very own marine mammal, Matt.
Thank you. Thank you. Wow, y'all. That was cool. Thanks for being here. Thanks for bringing a beautiful sunset as well. Can you all hear me okay? Okay, everyone online, can you hear? You good? All right, sweet. Excellent. Well, I have a confession to make. Please step with me into the confessional. as I make this confession, Father, for I have sinned, for I didn't know liberal arts colleges were a thing until I was like 35 years old. Until I started working for one, I didn't actually know what they were. And over the course of this last 10 years, I've fallen in love with these institutions and found a niche for myself that I didn't even know existed. And I'm super excited to tell you guys about it tonight. This combination of being able to do research, applied research that then helps conservation for animals that I know and love, as well as involves people like yourselves out in nature, sharing my passion for nature, sharing my passion for these whales, and hopefully impacting the next generation to pick up the mantle and move on. That's my confession. Now let's get back to sinning. Just kidding. So yeah, Sarah mentioned this, I've been fortunate enough to work at a number of liberal arts colleges. I finally stuck at Ursinus, which is a small college outside of Philadelphia, with some distinguished alumni, including an F1 progeny of our very own director. Genetics jokes. Come on, geez, a bunch of anatomists in the room and programmers. So before I met this place, this was my happy place. So and it still is. I desperately love being at sea. I desperately love being on the front of a boat, tied in high seas, getting splashed and soaked and windblown, approaching whales, collecting genetic samples or attaching satellite tags or what have you. This is where I feel like I really thrive. And this love for me started when I was even younger than you all. So this is me. Hard to believe, right? Only a wee little beard at the time. So this is me back in back in the last millennium, 1998. Who was born in this room in the last millennium? Oh, wow. Jeez, y'all are whippersnappers. So little. So this was my first real research job. And guess what, Sarah Morris? It was a bird job. I studied greater prairie chickens, an endangered species, on the tallgrass prairie of Oklahoma, about 45 minutes from where I grew up. But yeah, this is before my freshman year as an undergraduate at Oklahoma State. And then I was really fortunate enough to make my way into research programs as an undergraduate at Oklahoma State, including the very first black bear study in Oklahoma. There's only a very small range of black bears in the southeastern corner of Oklahoma in the Ouachita Mountains. And we studied the heck out of those bears. We even put some to sleep. That's a sleeping bear, not a dead bear. And that bear was so big, we couldn't weigh it with our scale. So it maxed out the scale. We estimate it's about 450 pounds. Its head alone weighed probably 100 pounds. Just getting that onto my lap was quite a workout. So that was fun. And then I have a funny story to tell about keeping your options open. So I had done some really cool work, been very fortunate to get involved with a lot of ecological studies and considered myself a field biologist. I took genetics. Anybody taking genetics? Okay. Yeah. So I took genetics as an undergraduate. This is the fall of my junior year. I had a wonderful professor who I dearly loved, but I was terrible. I was like scrapping to make a C in that class and was not happy about it. And this professor, I started chatting with this professor, maybe hoping to get slight bit of extra credit, maybe put me up into the B- range. It turned out the professor he trained dogs. He was a hunter and he trained hunting dogs. At the time, I was doing some part-time work just to try to get some play money at a training facility as well, dog training facility, separate training facility. So we started talking about dogs. We ended up hitting it off. He said, Matt, you should come work in my lab. And I said, genetics? You grade my exams. You know how badly I'm doing in this course. Why do you want me to work in your lab? He's like, I think you're cool. I like you. You seem to be interested in wildlife. I'm interested in wildlife too. He introduced me to this concept of conservation genetics, applying genetic techniques to endangered species or threatened species that need our help. And I started volunteering in this lab. I started learning how to pipette clear liquid from one tube to another in very, very small volumes. I learned in that time how to extract DNA, how to sequence DNA, how to genotype individuals. I did my undergraduate research on phylostomatid bats, and never planned to be a gene jockey, ever. But those techniques allowed me to go on and do other things. So it opened a whole new opportunity to do conservation genetics on whales, something that I was very passionate about since I was a wee little lad. Growing up in Oklahoma, so many whales there. Really, really love those whales. But it certainly piqued my curiosity. So I leveraged those skills to work at the Natural History Museum in New York. Sarah mentioned that. I was at undergraduate RU for 3 months, and I stayed there for 7 years because I just kept going, kept rising through, and then ultimately managed the genomics facility there for several years. And during that time, I started doing a lot of work, not just on whales, but in this realm of determining units to conserve. So I'm going to read this quote, and then I'm going to dissect it for you. The Endangered Species Act catalyzed an era of science and policy meant to scientifically evaluate, quantify, and validate units within species upon which the ESA would act. So this is the Endangered Species Act of 1973. Lawyers wrote this, but they wrote it with the help of scientists who realized the fact that in order to conserve species, you actually have to conserve accurately the populations within those species. Okay, so another sort of definition of conservation units are groupings of organisms below the species level, species, subspecies, and populations, containing the biodiversity necessary for the generation of new species, right? The persistence of species following environmental change and local adaptation. So this is, as I said in my class, this is where the rubber meets the road in terms of natural selection and evolution. And we need to conserve those things, right? It's the Endangered Species Act, but it actually conserves things like subspecies and distinct populations as well. So this is really cool. I'm kind of trained up in how to do these things, right? So now I can start applying those genetics techniques that I had no interest in learning to all these open questions about how biodiversity is partitioned across different landscapes. It turned out through the whale work that we started to realize that marine mammals overall are highly undercharacterized. That just means that we traditionally think of all whales being all connected, right? The ocean is one big ocean. We call it different things, but it's all connected. Whales can swim a long way. They're probably all the same thing. But when we start to peel back the layers of genetic diversity, we realize that there's actually distinct populations, and in many cases, distinct subspecies, and in some cases, completely separate species. So there's a whole lot of work to be done sorting out and characterizing diversity in marine mammals. Well, that's cool. So after 7 years at the museum, I said, I either got to go back and get a PhD or I'll just stay in New York City for the rest of my life. I'm from Oklahoma. The city was getting a little old, so I decided to go back to school. I was fortunate enough to get a National Science Foundation graduate research fellowship. How you doing, Maddie? To go to Scripps. And I went to Scripps and studied these wackadoodle things. Notice anything weird about this dolphin? It's AI, hallucinogenics. No, it's a forward-facing dorsal fin. Yeah, so these are a strange subspecies of spinner dolphins, dolphins that jump out of the water and corkscrew and then splash back down, that exists off the west coast of Central America. So these animals were the subject of a huge amount of bycatch, so unintended catch, in a massive tuna fishery, and their populations were highly depleted. So they're still remain just a fraction of what they once were. So we were charged, as I did my work at a NOAA lab, and the NOAA lab was charged to figure out how many of these things exist around the world. So we tried previously to figure this out using a couple genes here and there, and we didn't find any differences. But you look at these things and you're like, those kind of look different, right? Any untrained fisherman can look at these things and say, they're different things, right? The skull morphology said the same thing. So my advisor spent his whole career studying the skulls of these things and labeled them all different subspecies, but there was no genetic evidence. So I applied some genomics techniques, sequenced a whole slew of these things, and it turns out that diversity is represented. It's just deeper within the genome than those few genes that we had looked at before. We also found some new diversity. There's an undescribed subspecies in Hawaii as well. So if anybody's looking for a fun paperwork job to do, you can describe subspecies in Hawaii. And then we found a new population off the coast of Mexico as well. So these are really important things to know if you're trying to manage the recovery of these groups, right? Where do you draw the boundaries? That tells you how you count them, right? So if your boundaries are big, you're gonna have a big population. If the reality is that they're actually very small, you may have a very, very small population. So we need to know what those boundaries are, and that's what genetics can do for us. I also was involved with some work on Brutus and Eden's whales. We'll talk a lot more about this in a minute, but this is fun stuff we did with colleagues all throughout the Indian Ocean and Western Pacific. These are very strange beasts we don't know a lot about. We published a paper in the early 2000s on this work, and it sort of just keeps going because it gets more and more interesting. While I was at the Smithsonian, I generated a whole bunch of genomic data looking at the sort of relationships among all great whales. So this is sort of like, kind of think of this as sort of a genome scan approach where we're not sequencing the whole genome, but we're sequencing a large, nearly 3,500 chunks of the whole genome. And then we're building these phylogenetic trees to kind of tell us what the relationships are. So a couple of cool things. Those top two species, Balanoptera brutii, Balanoptera edeni, have been lumped for most of their history as a single species with two subspecies. Well, our data are basically saying they're not interbreeding, and they haven't been interbreeding for a very long time. So it's very likely that those are independent species. So we're in the process right now of doing a little bit of that paperwork. The other thing is, anyone heard of Rice's whale? It's kind of a hot topic these days. Yeah, so this is our whale. This is the American whale. This is existing in our own backyard in the Gulf of Mexico. Exists nowhere else in the world, endemic to the northern Gulf of Mexico, and was just described in 2021. And to top it off, it's critically endangered. So there is 50 animals left. Yeah. That wasn't bad enough. What happens in the northern Gulf of Mexico? Say that again. Yeah, that's right. Black gold. Texas tea, they say. Yeah, there's a lot of that under that Gulf of Mexico. So there's a lot of conflict now, a lot of interest from oil and gas companies, politicians, things like that, about the reality of Rice's whale. Is it really its own species? Okay, so we included Rice's whale in this analysis, and you can see it coming out in the green, the third one down. Sure as heck to me looks like something different. Very independent of those two, Brutus and Eden's whales. Okay. Cool. In addition to that, there's a whole lot of mess even farther down into the tree, and we don't have to get into the details of this, but the family that includes all the large whales is not biologically accurate. It's actually paraphyletic, which means that there's actually a family of gray whales nested inside the family of Balanopteridae. So we got to figure that all out. We've got a paper in prep that's trying to do all this, but you can imagine the confusion. Same thing with the genera Balanoptera. It's also got stuff inside of it, so we got to sort all that out. We're probably gonna erect like 3 more genera just to like help sort it out. So there's a whole lot of taxonomic revision that needs to happen in here. We're gonna pull things out, separate them, and make it nice and clean so it represents the biological reality. Okay. So in addition to that data set, we generated some mitochondrial genomes, and the pattern is roughly the same. We also did a cool analysis. I got to credit Michael McGowan for this at the Smithsonian. He helped me with this analysis. But some sophisticated stuff where we can actually do divergence dating. So we basically take our tree and we calibrate those little nodes based on the fossil record. And then we can estimate the date when these things diverge. So the question is, for Rice's whale, okay, maybe it's something new, but it's got to be like something recent, right? Well, not exactly. So it turns out it's about 3 million years old. If you know something about your own species history, there was no Homo sapiens 3 million years ago, right? That's a long time ago in terms of speciation. Okay. Our hypothesis is that it was probably segregated by the rise of the Panama Isthmus, and that's what sort of helps drive the divergence. But it's been its own thing for quite some time. All right, the cool thing about this is we've got students working on this. So some of you met Lily Kratz, who's on the left here. She's actually just graduated. She's coming back out to Scripps as a TA. Last year, she actually, we brought a mobile genetics lab, set it up in PK, and she did some of these DNA extractions and quantification right here on the island. And then Aviance also just graduated, and she's been helping me out with the morphology. So as part of our taxonomic revisions, we've got to do some morphological analyses to be able to say, oh yeah, the skulls are quite different too, or here's where the skulls are the same. So Aviance has been working on that as well. Cool, so lots of work in these crazy medium-sized balanopterid whales. This brings us up to the title of the talk, which is integrating student experiences and experiential learning in the study of the tropical whales. So when I was at the Smithsonian, I was approached by this group, Panacetaceae, and they said, one of our board members is retiring. Would you come on as a board member? And I was like, that's cool. You guys do some cool stuff. You do like some research and some education and conservation stuff. Sure, I'll do that. It took me about 2 hours every year. Sat on a Zoom call. Sweet. Easy peasy. Until they started asking me to come out into the field. And I was like, no. Can't do it. I got other things. Finally, they drug me out to the field, and I fell in love with this field site. So they work in the Gulf of Cherokee, which I can probably show you the picture somewhere. Ooh. That's this Gulf right here. So to orient you, this is the Panama Canal is right here. Okay. And that's where Panama City is. And this Gulf of Cherokee is actually a giant holding site for giant ships. Okay. But you get over here, and the Gulf of Cherokee is just beautiful. Much of it is marine reserves. Coiba Island right here is one of the biggest islands in the Pacific, or on the east coast of the Pacific. And it used to be a jail. So it's like pristine. It's really cool. Anyway, it's a beautiful, beautiful spot. So I went out there with my colleague, Kristen, who is the director of Panacetaceae. She's a humpback whale biologist. Panama is really unique in that it supports, provides habitat for both the northern hemisphere humpback whale populations and the southern hemisphere. They migrate synchronously, so they don't really cross over much. They do this, okay? And so we're there mostly when the southern hemisphere population is there. That's our summer months, okay? And the southern hemisphere population is doing quite well. Talk about that in a bit. That northern hemisphere population feeds and breeds off of California and Oregon and Washington. They come down to Central America. They are not doing very well. Okay, so not increased post-whaling and still really haven't risen back. So most of the work that Kristen and her team did was population monitoring, where they were out there, they were taking photos of these animals, doing market recapture analyses, trying to understand population dynamics. So I went out there once with her, and I was like, this is beautiful, it's amazing, it's great. And she happened to mention the fact that occasionally they see Brutus whales. And I was like, no, you don't. You're lying. I had seen Brutus whales on 2 other occasions. One was in Oman, and the other one was in the Western Hawaiian Islands. And Brutus whales have no interest in humans. Like usually when you see them once, maybe twice, and then never again. They'll come up, hey, Brutus whale, how cool is that? Let's go see it. And you never find it again. Okay. Kristen had seen these multiple times in Panama. And I was like, yeah, whatever. Sure, Brutus whales, everyone says that, right? So I went out with her for the first time in 2019, and just flippantly said, maybe we should go look for Brutus whales. And she said, sure, let's go tomorrow. We went over where she usually finds them. Sure enough, there they were. I was like, nice party trick. Never happened again. We went back to studying our humpback whales. The next year it came out. I said, hey, let's go look for Brutus whales. She said, sure. So we went over to the islands where we find the Brutus whales. Sure enough, the bastards were there again, just waiting there. And I'm like, Kristen, I don't think you understand what kind of situation this is. In most places around the world, Brutus whales don't care about humans and they don't want to be studied. In this population, it seems like they don't care that much. So, as I said, they're pantropically distributed. Seems to be most places around the world, they don't migrate. You can see them all year round. But as I said, they're really, really hard to study. So nobody's really done much in terms of tracking. They hate attention. So they're mostly introverts. Really challenging to study. And there's probably a whole lot more cryptic diversity in these than we know. So aside from a couple of places, the North Pacific being one, the Japanese really like Brutus whales, so they killed a bunch of them, and the South Africans killed a bunch as well. So aside from those two areas, Brutus whales were not really targeted that much. Hence, we don't know that much about them. As I said earlier, the taxonomy is pretty uncertain. We still don't really know. They still don't really have accurate names currently, and very little information is available on their migratory movements. So after all my skepticism, Kristen and one of our good colleagues, Daniel Palacios, published this paper a few years back where they finally pulled together all the data, all these random sightings that they had seen through the years, and did a kernel density analysis. And that's what you see in the map on the right. And these hot spots, these hot, warmer colors, are where they see Brutus whales. Happens to be around a pair of islands called the Contreras Islands. Okay, and that, sure enough, is where we go every year, and they're there every June through August. They seem to really like that spot. Okay, you ready? Greg, fall in love. Just saying. These are beautiful creatures. Okay, the weather is not always like this, I should say. This is a very rare day. So calm, the waves don't even move. Okay. Oh, attacking my poor computer. So you can see how little of the body comes out of the water. Imagine slightly higher sea state. Really challenging to see these animals. Okay, let's see if we can.
Cool neck too.
Where's the cursor? There it is. Cursor. Come back. Okay. Nope. It's gonna go. It's gonna happen. Cool. So this next video that you're gonna see very shortly is collecting a biopsy sample of these animals. So yeah, so I was skeptical that we could actually even do this, right? Until we started getting weather days like this, where it became clear that maybe we could approach them. Maybe we could get some biopsy samples. Okay, really. Oh, okay. Sweet. All right, ready? Okay, so what you're seeing here is the same individual that we saw in the previous video. It's going to do a couple of the surface to take a couple of breaths, and then it's going to arch slightly to do a terminal dive. So it's going back down to its foraging depths to eat some yums. And you'll see me in the front with the crossbow in my happy place, with one of these modified crossbow bolts. Did it happen already? Did I miss it? Okay, cool. So he's about to come up. Like it's in slow motion. Here it comes. Okay, gonna arch. Boink. And that's all it takes. That's all it takes to get a little bit of skin and blubber. And from that skin and blubber, we can get all sorts of fun genetics. We can get ecology. That's the celebration. We can get stable isotopes. We can get contaminants. We can get fatty acids. We can look at hormones. We look at pregnancy and stress and things like that. We're also able to use genetics to identify the sex of the animals. Can't tell them apart in the field, but when we get back to the lab, we can do that. So that was in 2024. And then in 2025, we went back and we said, well, let's try to answer this question. Are these whales moving around, or are they really just sitting in one place? So we mounted an all-star team, including me and my happy place there. And we decided to, in addition to doing all the things we normally do, like collect genetic samples and collect photo identification, we're going to deploy some of these limpet tags. And these are 50-gram satellite transmitter tags that are implanted percutaneously, so they don't go very deep. They go about that deep into the blubber. We aim for the dorsal fin, so they're going into that cartilaginous bit. All the anatomy and marine mammal class actually saw that, right? We dissected the dorsal fins, all cartilage in there, and that's really tough stuff for the barbs to stick on. It also provides a nice platform for transmission of the satellite location. Okay, I just found out that baby gold chicks weigh about the same amount as a satellite tag. So we were able to deploy those in 2025. Some of our results so far, again, this project has been really ongoing for the last 3 years, but before that, sort of opportunistically, our catalog from photo ID includes about 35 animals. Recall that we only started doing this in 2025, so it's pretty early on. Over 11 days in 2025, we cited 13 individuals, and of those 13, 7 had been previously cited. So that's a really high recite rate. 3 of the whales have been seen twice before, and 1 of those whales have been seen 4 times, including over a span of 11 years. So all indications, although we haven't done a mark recapture abundance estimate, are that this is a pretty small population and likely insular to this region. We've collected 21 biopsy samples. 9 of those have been imported into the U.S., and those are the ones that Lily was working on. All 9 of those samples are male, which is an interesting finding. Could be just a small sample size. Could just be a bachelor pad hanging out. We've also sequenced mitochondrial DNA. I literally got this stuff back a couple weeks ago. I haven't looked at it yet. That's a screen grab from the data. And we're going to be importing 12 more samples this summer. These are our results from the satellite tagging. You can see the tags deployed there. And the far right column is the duration. So average, that was about 28 days for those tags to stay on the whales. And then they fall out naturally. So the barbs over time, so they just work themselves out, and then they sequence. So we got about a month's worth of data for these things. Are you prepared? See the results. Okay, so just to thanks, Nick. Thanks, Nick. So just to reorient you, we're looking at the Gulf of Cherokee, which is on the left. That's the province of Cherokee in the red color there. We can sail across that. Well, we can motor across it. There's no sails involved. We can motor across it in about 3/4 of the day. So for a whale, it ain't nothing, right? Our hypothesis was that they probably didn't move much, but in reality, they can go if they want to go, right? They can go up to Costa Rica, down to Ecuador, over to Colombia, to Galapagos, wherever they want to go. At least we thought they'd probably go over to the Gulf of Panama just to check it out. Here's the results. Are you ready? Okay. Ready? What? What? They're going nowhere. They're like sitting on the couch eating Cheetos. Like, what are they doing? It's ridiculous. Yeah, they're just staying in one spot. That whole month of tracking, they're just sitting there. All right, we even had one individual that we were kind of a little concerned about. Because this individual, so that's the Isla Coiba, and then just north of that where we tagged him is the Contreras Islands. And that guy didn't move more than 15 kilometers from where we tagged it. So we kept seeing the guy. He was alive. It's not like he was floating there dead or anything. But he just didn't move. Quite literally a homebody. He's got everything that he needs right there. Why would I go anywhere else? So pretty cool stuff. So Sarah mentioned I'll be going back on a Fulbright fellowship in the spring. The idea is to bring more of these tags and to see what they're doing during the dry season and see if they're still doing that same pattern or if they're moving around a lot more. Okay, I'm going to breeze through this other stuff because it's super fun, but not as fun as that other stuff. This is more about using Panama as a fantastic entryway into marine science and marine research and conservation. Panama, as we discussed, is a crossroads for these two populations of humpback whales. They're very abundant on these breeding regions while we're there. Easy to study, very, very outgoing and extroverted. And Panama is a real safe place to be. That bottom right one will show you, however, the shipping lanes going in and out of the Panama Canal. So this place is not without its conservation challenges. So another part of my Fulbright fellowship will be to build some relationships with Panama Canal Authority, their environmental ministry, some of the universities there. Smithsonian has a base there. To really try to build up some of this marine conservation work. Well, what I'm here to talk to you about is incorporating students in research. Okay, so these are two of my students in 2024. That's Nick on the left and Michaela on the right, checking out some pantropical spotted dolphins. This is Kate Lloyd, who's a rising senior. She came out with me last year. And these guys really cut their teeth on humpback whales because the humpback whales are so ubiquitous there. They're really easy to study. They're also a great conservation success story. So Southern Hemisphere stocks have all rebounded, so much so that they're no longer on the endangered species list. This is a little hello from one of our humpback whales who swam right underneath the boat last year. Kate got this great video. I did that just to slow things down a little bit because I was going too fast. There we go. Cool. And so, as I mentioned, Kristen, my colleague, started this humpback whale monitoring project about 25 years ago. It's a really long-standing time series that we're excited to keep going, especially this year. This year is supposed to be a massive El Nino year. So it'll be interesting to see how the humpback whale population responds to this. Mostly what we do is collect tail fluke information. So we take photographs of the underside of the tail flukes that are like a fingerprint. And we can track those individuals along their migratory path and over many years. There's over 1,000 whales in our catalog. And our results are supporting the hypothesis that these guys are almost fully recovered, almost reaching their pre-whaling numbers. Sadly, our northern hemisphere population is not doing as well. Similar to our Brutus whales, we collect genetic samples, and we're working with a group at UC Santa Cruz to do hormone analyses. Some really interesting studies about how hormones change throughout the migratory pathway. So this group in Santa Cruz works down in Antarctic Peninsula, where these animals go to feed during their feeding season. And we work at the end of their migration on the other side. So how do hormones change as they go through these massive thousands of miles of migration? Both sex hormones like progesterone and estrogen, and also stress hormones. Remember, these animals are fasting the entire time they're in Panama. We also collect bioacoustics. We collaborate with a group at the University of Vermont, Laura May Collado, who does some really great work. She's from Costa Rica, and we send all of our recordings to her. We're also looking at behavior and energetics of moms and calves. The highest behavioral category of humpback whales we get there are mothers with calves. And so they're really easy to study. because they hang out in these shallow lagoons. They don't move a whole lot. So we've invested in some of these suction cup tags to look at what their behaviors are and what their energetics are. So how much are they moving? How much are they diving? What are they doing? So let's see if this works. So this is us deploying our very first suction cup tag. It's on the end of a 6-meter pole. So it looks like a pole vaulting pole. It's made of carbon fiber. And as you can see here, Dr. Leslie gets a little bit too excited and drops the tag. Whoops. That's an epic fail. But as you know, that's how science works, right? We do things, we fail, and then we learn from those things, hopefully, and do it better the next time. So this is the next time. Okay, you see we're all kitted up. We've got our helmets on in case that pole kicks back and brains one of us. It doesn't hurt us. You can see we're trying desperately to stop it from flopping so much, but it's a little swelly. That mama's kind of moving away from us a little bit. So we're going to give her one more surfacing. Going to come up and dive. And as she does that big humpback arch, the video will stop. Oh, here we go. Yay! So on these video or on these in these tags, there's all the sort of same stuff you have in your cell phone. So things like accelerometers and gyros and GPS tags, and there's also depth, barometric pressure depth gauges. There's also a video camera on the front, and there's also hydrophone. So we can hear them, we can see what they're doing, and we know where they're going and what they're doing. Okay, cool. And so we can get data like this. So this is a dive profile from a mother humpback whale. Our hypothesis with humpback whales, when they're moms, they stick really close to the surface so their babies don't have to go very far. But these data suggest otherwise. This animal is diving down to 30 meters quite happily, multiple times over this short period of the tag deployment. The video cameras give us amazing insights into what they're doing, how they're interacting. We're actually planning on tagging some of the calves this year so we can watch how often they suckle. This is a calf that's riding between the mom and the son, basically. Really, really cool video. Okay. In addition to that, we're doing some drone work. I published some papers on using drone photogrammetry, so the ability to measure whales from the sky, using some commercially available drone technology, plus some modified altimeters. And it actually turns out to be a really simple trigonometry problem to collect data and then extrapolate or measure whales along the surface of the water. So this is my very first student, Colin Perkins, who came out with me in 2020, just before the pandemic. And we've published 2 papers now together. And this is the data that we get. So these are amazing photos of the mom and calf. But really what we're interested in is photos like this, where the animal's flat on top of the surface. And we can use these images, counting up the number of pixels from tip to stern, and then using those calculations to measure the whales. In addition to measuring the length, we can measure the girth, which is kind of cool, because then you can model how fat the whale is. Okay, so what we want to see are big fat whales. Okay, so it'd be really interesting to do this post El Nino to see if that's what we in fact see. These whales are quite healthy. Cool. I don't know why this is in here, but oh yeah, I do. Okay, so this is all really cool stuff, right? That's incorporating undergraduates. They're helping out, doing all this work. I used to give this talk where I would, it was like a job talk or whatever, where I was like, this is my approach. I go out in the field and then I do some geneticsy things and then I analyze some data. And then I go take this to places like the International Whaling Commission and talk to managers about this and get recommendations. And then I go back into the field and do more analyses and stuff like this. But what's cool about being at a small liberal arts college is that now my students can do this as undergraduates. So they're the ones now who are deploying tags and taking photographs and doing the genetics. And in even this last year, Nick and Kate were able to go to the U.S. Marine Mammal Commission meeting and participate, or at least observe, how science gets incorporated into policy action. So pretty amazing stuff. I'm super excited about this. Sarah already mentioned it, so I don't have to talk about it. But I'll be based at UMIP, which is the International Marine University of Panama. And then I'm going to leave you with a couple of quotes. This one's by Carl Safina. I'm drawn to the wild, not because it is wild, but because it is sensible, logical, ordered, stable, and resilient. Wild nature is everything we're struggling to regain. And then this one by Richard Louv. Does anybody know Richard Louv? He wrote Last Child in the Woods. He also wrote Vitamin N about nature. Really, really good author, really interesting stuff. The future will belong to the nature smart, those individuals, families, businesses, and political leaders who develop a deeper understanding of the transformative power of the natural world and who balance the virtual with the real. The more high-tech we become, the more nature we need. And that's why I love the fact that we're out here doing this type of stuff, using some really rad technology, but we're doing it in a setting that, I mean, look at that. Just turn around for just a second. We are really, really fortunate, folks. It's a cool place to be. Okay, I'll just end by giving a shout out to some other of my students that aren't Marine. This is some work that these two students, Britt and Ryan, have done working on campus, studying bat diversity and activity using little acoustic recorders, audio moths. So Britt just defended. She was a distinguished honor student last year. And then Ryan and I just went to the American Society of Mammalogists meeting 2 weeks ago. And that Ryan's presenting the information there. Okay, and I'll leave you with a couple of take-home messages. I couldn't have done this at all without mentors. Mentors are so important. Okay, so keep them, listen to them, and never stop looking for them. I made a mistake when I was in graduate school to think that my major advisor was the only mentor that I needed. That was a terrible mistake because he was great for the geneticsy stuff, but he didn't get me in terms of the field stuff. Okay, so a diversity of mentors is really helpful. As you saw with our suction cup tagging experiments, failure is okay. Okay, life is a science experiment. It's okay to fail as long as you collect the data and reflect on it and then try it again. Okay, and then remain open to serendipity and change. I mentioned my story about bat and genetics. I had wanted to have nothing to do with sitting at a bench pipetting clear liquid from one tube to another. But it turned out to be a really useful tool and remains in my skill set today. And then finally, this is more advice to myself than anything else. Just keep going. Sometimes it's hard just to keep going, right? Okay, so back to my happy place. This is my place, but I've developed a whole nother happy place. And that involves my students and watching them thrive and getting them active and involved in the stuff that I love. And that's where I'll end. Thank you all.
Thanks, Matt. We do have time for some questions.
Hello, that was like a super, oh, this is not on. That was a super good talk.
Oh, sure.
Super sweet how you shout out your students and all that, and a lot of interesting research. I was wondering, like, you referenced as one part, like, how, like, taxonomy is still arguing about how to categorize these things and, like, how many new species or genera, and that this is a process and that they'll probably come up with a bunch of new things soon. In the next few years, what do you think is the situation might look like with that? Like, how many, like, what do you think it'll look like in terms of genuses and species, or how much will it change really?
In terms of Balenopterids?
Yeah.
Yeah. So just a little bit of backstory. Balenopterids are currently all within the same genus. They're currently all within the same family. We have a unique family for things like gray whales, which are a little bit weird. They stick their faces in the mud. They have strange, like, cranial morphology. And so they've been listed something separate. The humpback whales, they have those big white flappy arms. They've also been characterized as a different genus. Our data are basically saying that they're all within the same clade. So there's no reason to have Megaptera separate unless you're going to name everything else separate. So people get really upset about changing taxonomy, and some people really love gray whales, some people in the taxonomy world, and humpback whales. And so they don't want us to see all of that subsumed into one genus. And so what's most likely going to happen is that we're going to name each of the subclades their own genus. People will hate that too, but what are you going to do? With that? Do it anyway.
Hi,
Hi.
thank you so much for that presentation. It
You're
was
welcome.
incredible.
Thank you.
You mentioned a couple of times that the southern population is doing a lot better and the northern isn't. Do you think that has something to do with like global shipping or sort of like population, like human interactions in the ocean? Or is there potentially something else going on?
So the smart answer is we don't know. There's a lot of really smart people trying to figure it out. We just don't know. I doubt that global shipping is a problem too much. So Panama is sort of it's probably the hotspot in terms of global shipping in their range. Yeah, the Bay Area too is within their range. So there might be some overlap with that. But we don't tend to see a ton of humpback ships route. And Panama is the very end of their migration. So that's probably part of the reason we're not seeing very many in Panama. But it's death by 1,000 cuts out there these days. So it's a real struggle. It's not clear, all the other populations are doing okay. Not quite clear what's happening. It could be some sort of effect of just being small, right? We know that becomes an issue. Once you get small, it's hard to get back up. But yeah, it's a whole lot of unanswered questions.
Do you foresee any possible genetic changes in whales as they attempt to adapt to climate change?
Ooh, that's a good question. Most likely there will be. It's going to be hard to tell because those things take a long, long time to improve. But I would assume yes at some point, but it'll be after my lifetime for it. Yeah.
Do you have any hypothesis about why the range of that species that you're studying is so small? Are there any other closely related species that have a similarly small range?
Great question. Great question. Yeah. So what we think possibly is happening, and I've got a grant about this that I really should resubmit. It's really interesting. But what we think is probably happening, with those analyses where we can date the divergence. So there's a whole clade of animals, including Brutus, Edens, Rices, Omoras, that appear to be kind of this tropical, subtropical, non-migratory group. Sadly, we don't know much about any of those, but they seem to latch onto these sort of small to mesoscale oceanographic features and just like hang out. So Rice's whale, for instance, hangs out in the northern Gulf of Mexico. And right now, its core distribution is around one canyon off the coast of Florida. It seemed to get all it needs right there. So it's quite happy foregoing this huge migration to go somewhere else because it's somehow making a living doing what it's doing. So it's not alone. Omora's whale does the same thing. So a good buddy of mine, Sal, who was the one helping us with the satellite tagging work, he's right there. So he's holding the rifle in the middle of the boat. So Sal studies Omora's whales in Madagascar, and his whales do the same thing. They've got these oceanographic features, some upwelling or canyon or something, and they just continue to snack there. Yeah. So we think something probably happened in oceanographic conditions in that time where they diverged from the other migratory whales, because the ancestral state of these large whales is the migratory state. So those guys are doing something different. We don't know what it was at that time, but it's a really interesting area of research. somebody should give me money to study it.
I was wondering if you could talk a little bit more about your process of bringing your research to policymakers and what kind of challenges you've been having.
Oh, absolutely. Great question. Really great question. So yeah, I'm involved in a number of things. I mean, Sarah mentioned the International Whaling Commission. I haven't been back there in a while just because of other responsibilities, but that's one area. So the scientific D of the IWC is a collection of probably the best whale biologists in the world. And they meet they used to meet every year. Now they meet every other year. And they basically take all the new science, they digest it all. They have little like to-do lists where one year they'll study North Atlantic right whales, the next they'll study something else. And they produce a report that then goes into the managers at the IWC. The managers are largely political appointees, but they have this whole big report so they can make decisions on that. Not a whole lot of whaling that happens these days. So much of the policy work that I do that makes a real impact is in other areas. So I sit on the NOAA Science Review Group for the Pacific region. And that scientific review group is a group of 8 or 10 of us that reviews every stock assessment that NOAA puts out regarding marine mammals. And so that's an area of service that I really love because it allows me to, collaborate with these great colleagues, but also tell NOAA, sorry, this Central American population of humpback whales is not coming back. We need to do something better. So those are ways. I also mentioned the Marine Mammal Commission. That's an independent commission that sits in Washington, D.C. that does all things marine mammal. And they have a panel of scientific advisors too. They have an annual meeting. I mentioned I sent my students there as observers, but I've also presented to them as well. So yeah, those are a few things. I will say, maybe we can talk offline about Rice's whale because there's a whole lot of shenanigans going on with Rice's whale right now. And I've served as an expert on some litigation for Rice's whales. And I think I could speak about that. Whoops, maybe I shouldn't have. No, it's okay. So yeah, some lawsuits that are coming down the pike about the federal government's handling of Rice's whales. And plus publishing op-eds and doing sign-on letters and all that kind of stuff to build public engagement with Rice's whale as well. I consider conservation biologist, these are things that it's important for me to do. And back to small liberal arts colleges, it's great because small liberal arts colleges are constantly like poking the bear, right? Like they're okay with we don't get any funding from the state, so let's just poke the bear a little bit. We're so small, nobody's going to find us. So yeah, my deans and the general counsel generally support me in speaking out against this kind of stuff.
Hi, thank you so much for that talk. It was really, really awesome. I really love the pictures. It was really, really great visual. I am so sorry if I missed it at the start, but I'm curious if you could talk more about, you said you had grown up in Oklahoma. So why whales? How did you get there?
Why whales? Why not whales? Yeah, it's funny because the previous Rock Talk, Tom Roberts had a very similar question. How did you get into this? And his answer was, I was a nature kid. I was an outdoor kid. And that's kind of my answer too. I just never, my dad was a computer programmer and I saw him work like this all the time. I was like, I don't want to do that. I want to be outside. Okay, so what do I do 9 months out of the year? Right? But yeah, so that's part of it. It's just, I had very active parents who were willing to let me, run around our 20 acres and, be our little game ranger and, study all the things. And then, yeah, I was a water baby too. It got so hot in Oklahoma during the summer. The best place to be was underwater. And so I just kind of fell in love with that. And then when I was about 8 years old, my mom would take us shopping, right? We'd go to Walmart or we'd go to the Sam's Club or the Costco or whatever. And we were really annoying. And so she would send us to this is my brother and I, she would send us to the book section and go okay, pick out some comic books and just sit there and read. Well, I found this coffee table book on whales, Jacques Cousteau's Whales. And I just sat there enamored by all these beautiful photographs. And there was a whole chapter on whaling and there was all this bloody stuff with whales getting cut up. And there's all this really cool information. And so I have this distinct memory of when I'm 8 years old, I'm like shuffling up to my mom at the checkout line. I'm like, Mom, my birthday's coming up. Can you buy me this book? She's just put it in the cart. We got to get out of here. So that's part of it, just being curious and like being willing to stick my nose in the book and like, you know, so yeah, combination of things. Oh, goodness sake. This is a plant. I told her.
No, it's nothing bad, I promise. So you talked about how you got the suction cup tags and how you got biopsy samples. How do you get the satellite tags on the whales?
Great question. So there's two methods. Thanks, Kaya. For those of you playing at home, that's my daughter. So there's two methods. We can use the crossbow to do it. The crossbow is a little less accurate. It's a little more medieval, if you will, but not quite as accurate. The way Sal's doing it in that photo is with a modified CO2 rifle. So this is the same rifle that you'll see on National Geographic when they're out tranquilizing giraffes or whatever. It's a company specially makes these for veterinarians to, deploy, tranquilizers basically. Well, we modify it to hold one of our satellite tag. And so, yeah, it's basically like a paintball gun, a badass paintball gun, CO2 rifle. Cool. Nice.
So one more round of applause for marine mammal Matt.
Thank you. Thank you. Thank you.
If you weren't already jealous, crossbow, CO2 canister, it sounds like a great set of opportunities. Next week, we have Tom Good, who will be teaching in our MERM class, our Marine Ecosystem Resource and Management. He will be presenting Making Use of Marine Canaries in the Coal Mine. Join us at 8 o'clock on Tuesday. Thanks again for everybody being a great audience, and thanks again to Matt. Great talk.